Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Combined Science Biology Paper 1 Foundation Tier from 2022. Paper 1 comes from, is tested on 4 main topics. Topic 1, cell biology. Topic 2 is on cell organization. 3 is infection and response, and topic 4 is on bioenergetics, and there's a total of 6 questions, and the paper is out of 70 marks. So let's have a look at question number 1. Foods are digested before they are absorbed into the blood. Figure 1 shows organs in the human digestive system. So if I was to label the parts here, that will be the mouth. That's the liver. That's the stomach. That's the small intestine. Now we could label the others as well. That's the large intestine. That's the esophagus. What else? That's the pancreas. Where insulin is produced. And the anus. Right. So which organ is the stomach? Is the first question. Stomach is C. And what type of enzymes is produced in the stomach? It's protease. Right? Mainly in the stomach, the enzyme pepsin is produced, which breaks down proteins to amino acids. Now carbohydrates, example is amylase. That you find it in the saliva. And in the small intestine. That generally breaks down carbohydrates, break down carbohydrate to glucose. And lipase, again, that can be found in the small intestine and usually breaks in fats into fatty acids and glycerol. Which term describes the pH in the stomach? Now give one reason why the stomach is this pH. The stomach pH of the stomach acid is acidic. Right? Give one reason for it to be acidic, because the stomach produces hydrochloric acid. So here's the answer. Produces hydrochloric acid, or you could say that's the optimum or the best temperature for the protease enzyme to act. Which organ produces bile? Bile is produced by the liver. The function of bile is to break down fat into small molecules and to neutralize the acid. How does the bile help in the digestion of food? There you go. Right? It increases the surface area of fats. It doesn't make it more acidic, it actually makes it less acidic. So it's just number 1. A student tested different foods for the presence of protein, starch, and sugar. Draw one line from each food molecule to the reagent used to test for the food molecule. So, protein, let's do the easier one. Starch is iodine solution. Sugar is Benedict solution. And protein is pyruvate reagent. Now I could actually write down the results for it as well. So sugar, Benedict solution, it changes color anything from blue. The Benedict solution is actually blue in color. It changes to anywhere between yellow, orange, or brick red. Sometimes we just call it as red. The more sugar there is, more the color intensity is. And for protein, it goes from blue to purple, and iodine, it goes from, iodine is orange in color, it changes to blue, black, or you could say purple. Give one safety precaution a student should take when using Benedict solution. Benedict solution is an irritant, so you could wear goggles so that you don't get it into the eyes. Okay. Use a water bath to heat the solution or mixture. So because you will have to heat it, so make sure that you take care and wash any spills from the bench or on the skin. Table 1 shows the results for one food sample. Color after test. Benedict solution is red. Pyruvate solution is blue. Iodine solution, it's black. So that means if it's turning black, that's a positive result. That's a negative result. That is also a positive result. Benedict is for sugar, protein, and starch. So, which of the tests show positive results? Benedict's and iodine. So, Benedict's and iodine. That is number 2. Starch molecules are not absorbed into the blood from the digestive system. Give one reason why. Now, starch molecules are big molecules. Sometimes you can call them macromolecules. Starch is made up of glucose. So each of those circles is a glucose molecule, but starch is branched as well. So it's quite a large molecule. Let's say it looks like that. Now, because it's quite big, it can't go through the cell membrane, right? So if that's the cell and there's spaces for the molecules to go through, it won't go through the gaps. If it was to go through, it needs to be small molecules one at a time. So the answer is starch molecules are too large. You could also say starch molecules are insoluble. That brings us to the end of question number 1. Let's have a look at question number 2. Question number 2 is on the topic of bioenergetics. Bioenergetics is referring to photosynthesis topic and structure of the plant cells and functions and so on. Question 2. Figure 2 shows a section through a leaf. So let's label the leaf. That is the waxy layer, right? You can call this the upper epidermis. The palisade layer, this is called the spongy mesophyll, and lower epidermis, and X is the stomata, and these two cells which are either side, that is the guard cells, which controls the opening stomata so that gas exchange and water vapor can go in and out. So, and this is the air space, right? Empty space which is just filled with air. And this structure that you see is actually the vein which has the xylem and the phloem, and the small tubes is actually the xylem, and that's the phloem on the outside. Okay, let's have a look at the questions properly. Give one way that the palisade layer is adapted for photosynthesis. Now, how is it adapted for photosynthesis? The palisades layer, all of these dots, is basically chloroplast. Right? Compared to the cells in the spongy mesophyll, the palisades layer has a lot of chloroplast. Why? As light enters from the top, from this direction, when light's coming through, it's going to pass through these layer of cells. So you want the cells close to each other and pass through as many chloroplast as possible before it reaches the secondary layer. So the answer, cells contained many chloroplasts, is the best answer. Gases passes in and out of the leaf through small pores on the surface of the leaf. What are the small pores labeled X called? They are called the stomata. A student viewed a section of a leaf using a microscope. The student measured the length of one of the palisade cells. The cell image measured 28 millimeter in length. That's the image length. When viewed with a magnification of 4 times 400, calculate the real length of the palisade cell in millimeters. So they have given you the equation. Real length is equal to the image length, which is 28, divided by magnification is 400. 28 divided by 400 is going to give you 0.07 millimeters. Convert the real length of the cell from millimeters to micrometers. 1 millimeter is equal to 1000 micrometers. So how do you convert 0.07 millimeters? You multiply it by 1000. 0.07 times by 1000 will give you 70. That's 70 micrometers. Carbon dioxide can move into and out of cells. What is the process by which carbon dioxide can move into and out of cells? That would be by diffusion. Remember, there's 3 different types of transport mechanisms in plant cells and in animal cells. So active transport, that includes glucose, salts or mineral ions, like phosphates, nitrates in plants, and sodium ions. So in animal cells, osmosis is the movement of water. Water from dilute to concentrated solutions. Figure 3 shows a diagram of 4 cells. Each cell is surrounded by carbon dioxide molecules. So that's low concentration, high concentration. These are all carbon dioxide molecules. The concentration looks to be the same on both sides. And C, it looks to be the same on both sides. And D, it's high concentration on the inside, low concentration on the outside. Which cell will carbon dioxide move into the fastest rate? Now, which way will it move? It will actually move inwards by diffusion here. The rate at which it's going in is going to be equal to the rate at which it's coming out on both these cells, because the concentrations are the same. Here is going to be coming out. So it is going to be cell A. Reason, because there's a bigger concentration gradient, or you could say there's a steeper diffusion gradient of carbon dioxide inside and outside the cell. A student investigated the effect of different colors of light on the rate of photosynthesis. Figure 4 shows some of the apparatus the student used. So you have a beaker over here. Beaker, this test tube. Now bubbles of gas, that would be the oxygen. This is also oxygen that is collected here. Glass funnel. Right, the pondweed and the timer. So as photosynthesis happens, pondweed carries out the photosynthesis. It produces the glucose, which is going to be inside the pondweed, and the oxygen that is produced is actually coming out as bubbles of gas. The student placed the apparatus in blue light, then in green light, and then in red light. The student measured the rate of photosynthesis in each color of light. How do you measure the rate of photosynthesis? Well, you could count the number of bubbles in a given time. How many bubbles does it produce in 1 minute? Or you could find out what is the volume of oxygen collected on the top after, let's say, 5 minutes or 10 minutes. And then you can divide it by the total time to get it per minute. So what two measurements should the student make to calculate the rate of photosynthesis? For the rate of photosynthesis, you need to know the volume of the oxygen collected, or the number of bubbles, and you need to also measure the time taken as well. Give two variables the student should keep the same in this investigation. Now remember, you're only changing the color of the light. Now you want to keep the position of the light from the pondweed the same for all three different colors. The temperature of the water, that will affect rate of photosynthesis, and the concentration of carbon dioxide as well. So any two of these will be fine. Temperature of the water, the light intensity or the distance of light from the pondweed, and the concentration of carbon dioxide in the water. Table 2 shows the results. Rate of photosynthesis in arbitrary units. So let's say they're counting the number of bubbles. With blue light there was 9 bubbles, green light there was just 1, and red light there was 8 bubbles. Again, they are not saying the unit, so let's assume it's in 1 minute. So complete figure 5. Here they have given you a grid to draw a graph. What type of graph would you draw? They're asking you, you should label the y-axis, use a suitable scale, plot the data from table 2 as a bar chart. There you go. You are plotting a bar chart. Label each bar as well. So they've already given you the color of light on the y-axis. So what goes on the x-axis? What goes on the y-axis? You simply copy this bit. Rate of photosynthesis in arbitrary units, right? That comes from the table. So rate of photosynthesis in can you just copy part of it? I would just copy all of it just to make sure. Let me just check the spelling for that. Arbitrary, T-A-R-Y, arbitrary units. There you go. So how do you draw the bars for this? Remember, the blue is 9, green is just 1, and the red is 8. So I've got, if I count 1, 2, 3, 4, 5, 6, 7, 8, 9. There you go. That's 9. So if I go 1, 2, 3, 4, 5, 6, 7, 8, 9. Don't have to go up to 10. And let's plot the, for the blue is going to be 9. So let's see if I can get some straight lines here. No, it won't work. Hopefully this works. There you go. Faster. There you are. So that's for the blue. Green is just 1. Let me redraw that. So you might want to make sure that you're using a ruler for this in order to make sure that it's straight lines. So that's the green, and red is 8. That's red. Just to, okay, that's the blue, green, and the, you don't really need to shade it in your exam paper. And let's have a look at the next question. What color of light should be used to grow plants in greenhouses? Well, you want to use blue light so that you get the most photosynthesis because it produces 9. So that's blue. That brings us to the end of question number 2. Let's have a look at question number 3. Question number 3 is on the topic of infection and response. This question is about disease. Rose black spot is a disease where black spots develop on the leaves of rose plants. What type of pathogen causes the rose black spot disease? Now, if you don't know what rose black spot is, here's what it actually looks like. And I'm pretty sure you have seen plants, rose plants with leaves which look like this. And it is caused by a fungus. Plants with rose black spot disease often have yellow leaves. Suggest one reason why the leaves are yellow instead of green. Now, what gives green leaves its green color? That's chlorophyll. If there isn't enough chlorophyll, then it's going to appear a different color. It's not going to be green. So there is less chlorophyll or no chlorophyll, or you could say the chlorophyll has been broken down. So explain why plants with yellow leaves grow slowly. Now, you need chlorophyll in plants, in the leaves, for it to carry photosynthesis. Photosynthesis will produce glucose. Glucose, in return, is used in energy to produce energy, or release energy, I should say, so that the plants can actually make new things like cellulose, cell wall, make new proteins and all kinds of things, so for it to grow. So if the leaves are yellow, then that means there is less photosynthesis. You could, for 2 marks, you need to say there is less photosynthesis, or you could say there's less light being absorbed, so less glucose is formed or made. Question 3.4. The spread of rose black spot can be controlled using different methods. Draw one line from each method to control to the explanation of how it works. Remove and burn the infected leaves. What will that do? That would kill the pathogens. Water the roots of the plant only, not the leaves. If you water the roots, then it's not going to wash off the fungus elsewhere. So reduces the chance of pathogens being spread by water droplets. There you go. (3.5) Tobacco plants may become infected with a pathogen called TMV. TMV stands for tobacco mosaic virus. What type of pathogen is TMV? As the name suggests, it is caused by a virus. Now, what does that look like? Here's a picture of a tobacco mosaic virus infecting a tobacco leaf. So these leaves are quite big, right? And the yellow thing is mainly because of the virus on the leaves. Malaria is a disease caused by a protist. How is the malaria pathogen transferred to humans? How do humans get it? By mosquito bites. How can the spread of malaria pathogens be reduced? Now, if you reduce the amount of mosquitoes, then you would control the amount of malaria as well. So avoid sexual contact. No, that's not going to happen. Help cook food thoroughly. It's not passed through the food. Drain water from swamps. That is correct. How does that help? Mosquitoes actually lay eggs in water or standing water, dirty water, and those eggs will hatch into new mosquitoes. So if you drain the water from the swamp, then you stop the mosquitoes from reproducing. So let's write that down. Stops mosquitoes from reproducing. That's the end of question number 3. Let's have a look at question number 4. Cigarette smoking is the main cause of cancer in the UK. Mutations in cells cause cancer. What does mutations mean? Mutations are changes, or you could say damage to the DNA. Where in a cell do mutations happen? Well, you find the DNA in the nucleus. Why do some cancers develop into large tumors? Now, if it develops into large tumors, that's mainly because the cells never stop dividing. Right? The reason it's actually becoming into large tumors is because the cell never stops dividing. Now, cancer cells, you could have two types of cancer. So let's make a quick note of it. Cancer, first type is malignant. The malignant type can spread. Two is a benign, does not spread. Cigarette smoking has been linked to many different types of cancer. Lung cancer is the most common type of cancer caused by smoking. Suggest one reason why. Well, the cigarette smoke is going to go into the lungs first. So you're going to inhale the chemical first. So any of these points will give you the mark. Chemicals enter the lungs first, or the chemicals are inhaled, or you could say the chemicals are more concentrated in your lungs. A person with lung cancer can develop secondary cancers in other parts of the body. Describe how this happens. So let's say you've got cancer cells in the lungs, and when you're coughing, some of the cells might actually break away from the tumor and enter your bloodstream, and through the blood it can actually travel to other parts of your body, let's say in your throat or in your legs or wherever it wants, wherever it can go, and then it can stick in there and then become a tumor in there, start growing in there. So how does this happen? Cancer cells are transported in the blood. That's the main answer. Sometimes a person may need a lung transplant. The National Health Service will not offer a lung transplant to a person who smokes. Suggest one reason why. Now, if you're a smoker, unless you give up smoking, then it's going to be hard to get a lung transplant, because if you're given a second chance and if you smoke again, you're still going to damage the new lung, right? So, and there's limited amount of money in the NHS to treat people. So any of this, smoking will damage the new lung. NHS has limited financial resources, or the lung could be used for someone else who does not smoke, let's say a 5-year-old child rather than a 35-year-old man who's insisting on smoking. And the illness is self-inflicted, or the person is not attempting to help themselves. So why help? And it's true for those who have damaged their livers by drinking alcohol as well. So figure 6 shows data about skin cancer in males for different age groups in the UK. The data shows the number of new cases of skin cancer in 1 year. So you can see between the ages of 15 to 19, so every 4 years, right? As the age goes from 15 to 90 plus, the number of new cases of skin cancer steadily increases from 60 to 64, and there's a big jump from 65 to 69. 65 to 69 has the highest cases of skin cancer. And then after 69, it slowly drops, and then it's quite low at 90 plus. So you need to understand what causes skin cancer. That's mainly due to us being exposed to direct sunlight for long periods of time. So as we age, we spend more time outside because of work and we have our day-to-day activities, right? So if you haven't actually had skin cancer, then you're not going to develop new cases of skin cancer in your 90 plus, because the chances is going to be less. That's why it's fallen at 95 or 90 plus, because elderly people will not spend more time outside. How many more cases, question, how many more cases of skin cancer are there in males aged 40 to 44 than 15 to 19? So 15 to 19, how many cases do you find? Now there's 5 boxes for 100. So 100 divided by 5 is 20. So here it's 20. And 40 to 44, that's 301 box up. So I would say that's 320. So it's gone from 20 to 320. So the answer is 320 minus 20, which gives us 300 new cases. There are no new cases of skin cancer diagnosed in males younger than 15 years of age. Why? Why are boys under the age of 15 years old not diagnosed with skin cancer? Well, the amount of time spent in the sunlight is probably less, not enough for them to gain or get skin cancer. And when we are young, we also use a lot of sunscreen and so on in order to protect ourselves. So any of this answer will be fine. Little exposure to ionizing radiation, which is basically the UV light from the sun. Little exposure to sunlight or UV light. More use of sunscreen. Little exposure to carcinogens, meaning that chemicals like cigarette smoke and so on, which can cause cancer. And little damage or to the DNA. Give one conclusion from the data in figure 6. So there's a number of different conclusions. We could actually say any of this. Most new cases each year are in males or people aged between 65 to 69 years. And the new cases per year increases from age 15 to up to 69 years. So as you saw on the graph, it was going up. New cases per year decreases from 69 to 60 plus years. So those are the general trends in the graph. Survival rates for all types of cancers have improved over the last 20 years. Suggest 2 reasons why. Why are more people surviving cancer now than they were in 2000 or in the 1980s? Mainly because there's better drugs, better treatment. So any of this, improved treatment or drugs, earlier diagnosis, we can check for cancer better. Improved cancer screening, we can actually, doctors and GPs actually generally check patients whenever they visit, when they're in a certain age group. Improved patient doctor knowledge of dangers and treatments. So patients are more aware now to what to look for in order, let's say for example, lumps in their breast to detect breast cancer and so on. That brings us to the end of question number 4. Let's have a look at question number 5. Bacteria. Question number 5 is again on the infection and response topic. Bacteria can cause a variety of disease in humans. What are 2 similarities between a bacterial cell and an animal cell? A bacterial cell and animal cell will both have a cell membrane. They don't have cell wall. That will be bacteria and plant cell. Both have a nucleus. No. Animal cells have. Bacterial cells don't have a nucleus. Remember, they are prokaryotic. Both have cytoplasm. Yes. And both have plasmids. No. Bacterial cells have plasmid. Animal cells don't have plasmids. Salmonella food poisoning is caused by bacteria in food. Give one symptom of salmonella food poisoning. Do not refer to vomiting or diarrhea in your answer. Well, those are the common things that we know of when we talk about food poisoning. But generally, we do get a fever, abdominal or stomach cramps as well. What is the name of the first antibiotic developed? And that would be penicillin, discovered by Alexander Fleming. A child with several bacterial infection or severe bacterial infection was given a course of antibiotics. Figure 7 shows how the concentration of live bacteria in the child's body changed when taking the course of antibiotics. So, course of antibiotics was given at day 0 here, and the antibiotics didn't start working yet. So the number of bacteria, concentration of bacteria is increasing right up to about, let's say, 3.8, right? And after a day, just before, right, the 1 day is completed, it starts to go down, and then it steadily drops after about 4.5 days. The concentration of live bacteria in the body continued to increase after taking the course of antibiotics. Why? Well, it takes time for the medicine to be absorbed from your stomach into your bloodstream and to travel to where the bacteria is. So, any of these points will be fine. Only a few bacteria were killed, so live bacteria continued to reproduce. So not a lot of bacteria had started dying because of the antibiotic. Now or you could say there's a time delay before the antibiotic reached the bacteria or before it killed the bacteria. After 3 days of taking the antibiotic, the child felt better. There were still bacteria in the child's body. Why did the child feel better? Bacteria had become immune to the antibiotic? No. The child has become resistant to the bacteria? No. There are fewer toxins in the body than at day 0. That's the answer. See, it's the toxins which makes us feel unwell, either gives us the diarrhea, the fever, the stomach cramps, and so on. Suggest why doctors do not give antibiotics to patients with minor infections. Now, just because you have a small infection, if you ask for antibiotic, the doctors will not give. Why? To reduce and to prevent resistant strains of bacteria from developing, or you could say to prevent antibiotic resistance in bacteria. Figure 8 shows the blood viewed using a microscope. Right, let's see if we can label this. That's a red blood cell. And the large one is the white blood cell. Yes, it doesn't look like a white blood cell, but the black area is actually the nucleus. Right? White blood cells have a large nucleus. There are two types. You could have white blood cells with this large nucleus, or you could have white blood cells. It doesn't really have a proper shape, and what we call a lobed nucleus. So it basically looks like as if it's broken up. These are sometimes called phagocytes. These are memory cells. Now, these are also white blood cells, by the way. So C is the plasma. That's a liquid part, which is mainly water, mineral ions, a bit of salt. And D is broken cells, which we called platelets, which help in the clotting of blood. A vaccine will stimulate the production of antibiotics. Which part of the blood in figure 8 produces antibiotics? That would be the white blood cell, that is B. And which part of the blood in figure 8 starts the clotting process? That's the platelets, that is D. That brings us to the end of question number 5. Let's have a look at question number 6. This question is about cell division. Write the biological structures from the box in the correct order of size from the smallest. That would be the gene first, then the chromosome, then the nucleus, and then the cell finally. Figure 9 shows how a fertilized egg cell can produce specialized cells. So you've got fertilized egg cell, which divides by mitosis. Right, let's write that down. Divides by mitosis to form the embryo, and then it divides by process A to make red blood cells, muscle cells, and nerve cells, and all the different types of cells. Name process A, that is cell differentiation or simply differentiation. How many cell divisions are needed to form a 16-cell embryo from the original fertilized egg cell? So you start off with one cell. After one division, it becomes 2. 2 becomes 4. 4 becomes 8. You're multiplying it, right? And then 8 becomes 16. So that's your first division, second division, third, and the fourth division. So you need 4 divisions. In humans, a fertilized egg cell contains 23 pairs of chromosomes. How many chromosomes will there be in each of the embryo cells? So if you have 23 pairs, that will be 46 chromosomes. So how many chromosomes will be in each of the embryo cells? It will be 46. Figure 6 or figure 10 shows a cell cycle for a human embryonic cell. So you've got the start of the cell cycle, goes through stage 1, and stage 2 is mitosis, and stage 3 is not labeled. Describe one change in the cell that occurs during each of the stages of the cell cycle. You need to know what happens during each of the stages. So stage 1 is where the cell is actually just growing and doing its usual job and trying to get ready for mitosis. So you could say stage 1, the cell growth, it increases the number of subcellular structures, the DNA replicates, the chromosomes double, or you could say duplicate or replicate, any of them is fine. Stage 2 is for mitosis, remember. So that's when the chromosomes split apart or pulled apart. So one set of chromosome is pulled apart to each end of the cell, and the 2 nuclei is formed. Stage 3 is when the cell actually divides. You call it cytokinesis. The cytoplasm or the membrane divides to form 2 identical cells, or we can write it as bullet points. Next part, cell division is important for the growth of multicellular organisms. Figure 11 shows the mean height of boys and of girls from birth to age 18 years old. So the mean height at 0 age, the mean height for girls is slightly lower and for boys is slightly higher. So 20 for 5 boxes. So if I say 20 divided by 5, that gives me 4 centimeters. So boys are starting at 62 centimeters. Girls are starting at 60. So the girls are slightly shorter than the boys. Now, the girls tend to be shorter all the way by about 4 centimeters. And that's not 62, is it? 64. So all the way up to about 6.5, 7.5 to 8 years nearly. Let's say 7 years. That's 7 years here. So right up to 7 years old, the boys were always taller than the girls. And the girls slow down a bit more, and the boys actually keep the same pace. So now at this age, see where it's actually meeting up. Let's say that's about 11 years old. At 11 years old, both the boys and the girls are the same height of about 140 40. So the girls actually catch up. When they are 10 years old, they actually speed up their growth. See that line which is going up? So you could see that line is going up. The girls are actually growing faster between the ages of 10 to about 14. Now after 14, they're stuck with that same height, right, of 164 centimeters maximum height. Boys, on the other hand, they grow slowly. After 11 years old, they're not picking up the pace till about 13 years old. Now at 13 years old, their growth is quite fast. So you can see the growth is actually increasing really fast, and they're actually growing up to about 17 years old. 17 to 18 years old, boys are actually growing, and the maximum they reach is about 180 centimeters. Now, what I've just described is probably and definitely the answer to the question that you're about to see. So, compare the growth of boys with the growth of girls. That's your question. Use data from figure 11 in your answer. So, 6 marks. You could literally describe the way I describe it. You could basically start off by when they are born. Boys are slightly taller than girls. By how much? You could say 4 centimeters. And you could say right up to 7 years old, boys are always taller than the girls, and then you can talk about what happens at 11 years old when they're the same height, and between 11 to 14, girls actually grow faster than the boys, and boys actually start growing faster from 13 years old to 16 years old. So the answer that I'm going to give you is quite detailed. You don't really have to write all of this, right? But you'll see that you can pick out most of these points as you read through. What you need is about 6 to 8 of these points. You want some general comparisons, and you want, because they're asking about compare the growth of boys, so the rate comparisons. Rate is when you're saying how fast, right? Rate of growth is similar up to 10 to 11 years old. Girls grow faster than boys between 10 to 11 and 14. Girls have a greater increase in height between 11 and 14. So pause the video and have a look at the points. Right, you can pause this point for the general comparison, and for the rate comparison, you can pause at this point. Question number 6.7 is the last question in this paper. Give one way that cell division by mitosis is important in fully grown animals. Well, that's an easy answer. It's to repair of tissues or to replace dead or worn out cells. That brings us to the end of the video and the question paper as well. Thanks for watching. Hopefully you find this useful. See you in the next video.